So, hello everyone. Um, Welcome to the UK Human Data and Technology Club, which is convened jointly by ELIXIR UK and HDI UK. So, my name is Tim Beck, and I co-chair the club with Saskia Lawson-Tovey and Wendy Bacon. And so, this is the meeting that covers knowledge exchange on human and health data research infrastructure and technology. So if you've joined this meeting before, then welcome back. If you're new to this meeting today, then we have the opportunity now at the start of the meeting for you to introduce yourself. So if you would like to, then please go ahead, unmute, and feel free to introduce yourself. So this month, our focus is going to be on ELIXIR activities. So we alternate between ELIXIR and HDI UK activities. And this month, I'm delighted to welcome Professor Aedín Culhane from the University of Limerick and the ELIXIR Ireland node. Aedín is active in the ELIXIR cancer data community and is going to present an overview of Ireland's federated cancer data programme. So I'll hand over now, and Aedín, the floor is yours.
Thank you so much. Thanks, Marianne. I'm in the University of Limerick, which is in the southwest of Ireland, and I lead a cancer centre, the Limerick Digital Cancer Research Centre, and also an All-Ireland programme which is co-led by ourselves and Queen's University Belfast, called the eHealth Hub for Cancer. And I'll explain a little bit about those and how we're trying to bring cancer data together on the island of Ireland to really advanced data-driven research in cancer. And so let me just make sure that I am okay. So I moved back to Ireland in 2021 from Boston, where I had been at Dana-Farber Cancer Institute for probably over 15 years. Prior to that, I spent 7 years in the UK. And the cancer centre we've set up spans over 40 research groups and is really focused on kind of the technology side of assay development, sort of biofabrication, and we're doing a lot of spatial omics, and then increasingly working on kind of real world data and digital trial data. So we're not a classical, biological research centre. We're building on, the university is quite a technological university, and we're building on its strengths. And my own background has been in computational biology, bioinformatics, in clinical oncology for quite some time, supporting both kind of phase 1 and phase 3 trials, and also then looking at lots of different single cell and spatial omics methods more recently, and in probably like multi-omics data integration and tensor factorization and matrix approaches for bringing lots of different data types together. One of the projects that I'm not going to mention, and I probably should be mentioning, is kind of our role in Bioconductor. I'm on the governance board of Bioconductor, and I've worked very closely in the leadership of Bioconductor. A lot of the leaders in Bioconductor are in Boston. And we run through Limerick the global community engagement for Bioconductor. So Maria Doyle is in my team, and she's running a global train the trainer program at the moment. We're doing a lot of outreach in Africa and East and West Africa. We have about 70 trainers trained in the Carpentries Bioconductor program and are delivering teaching through that. We also recently recruited Nicholas Cooley, who is the developer manager. So this is really, how do we teach developers to develop better code? And within, like, because Bioconductor is an R project, a lot of it is working at how do we develop R and Python integration? How do we actually, can we create native GPU native R? So CUDA libraries that work better with R. Can we actually think of how we can do AI in R better, and then also like make that link between R and Python as transparent as possible. And if you've worked in R, worked in Python, it's much easier to work in Python from R than it is R from Python. So I think the R community kind of naturally goes between R and Python a lot more so than the reverse. So there's a lot that we're thinking of in that space. We also have monthly seminars running in that. I'm also head of Node of ELIXIR Ireland. I took over that job more recently, mostly, and we're trying to build a funding case for expanding ELIXIR in Ireland. And then I'm working on a lot of data infrastructure and standards, which I'll talk about. And so really it's how do we actually, make use of the fact that we have more and more data to drive biology and biomarker research. These are just a couple of papers. I'm not actually going to go into the actual science, though I probably should be doing, of the work that we do in kind of spatial biology, but a lot of it more recently has been looking at single cell and spatial biology to support immune oncology research and specifically CAR T cell therapy in kidney cancer. And then we've been doing a lot of work on kind of better methods for doing dimension reduction, which is the foundation of a lot of these work where you have these vast data sets and you're trying to bring the data sets down to a smaller scale or integrate across data sets. You end up using dimension reduction, sort of principal component based approaches. And that's a lot of where my research lies. And I think, the success of bioinformatics, particularly in cancer research, is really, really evident. If we look at all of the new drugs that are now being approved and actually making their way to patients, we see that 80% of new medicines are molecularly targeted. So, way back in the 80s and the 90s, cancer drugs just hit like a cell cycle, and it was a real kind of hammer approach where drugs weren't targeted. And increasingly, we're targeting drugs. By targeting drugs, we're reducing side effects. However, increasingly, access to those diagnostics is actually a barrier in cancer. And this is like some of the stuff that we've been thinking of a lot. And this is a quote from a Nature Medicine review in 2022. And it's like, we're getting more drugs that are actually molecularly targeted, but the paradox is then patients can't access these. And so, as somebody who's worked in a hospital, at Dana-Farber for many, many years and seen the benefits of when patients can actually access those drugs, and then seeing those gaps in access in the public health systems, that's one of our big areas that we're actually interested in. We looked at medicines access in Ireland, and this was a project with colleagues from Boston. So this was with Ali Van Allen's lab, and Brendan Reardon, who works with Ellie, is actually a PhD student. And so we wanted to understand what drugs are available in Ireland, but also then build platforms so that patients could quite easily see what medicines were available to them, and also then access to clinical trials, which is obviously really, really important, and particularly for managed entry agreements where we can get patient and medicine access to patients. So within Boston and in the Farber, we have this thing called Molecular Oncology Almanac, which is an open source knowledge base of gene-drug interactions, or gene-drug indications, sorry. OncoKB at Memorial Sloan Kettering is a similar tool, but the one at Dana-Farber is actually not, is under a full open license. So what we did is we actually went through all of the gene drug indications in Ireland, and we actually systematically put them into adjacent standard in GA4GH standards. And so we now have an Irish implementation of this. And this is really important for us because we now then have a reporting tool which will actually take this knowledge base, it will take a VCF file, it will annotate it, and then it will output a report that can be used in the clinic to guide precision oncology decisions. Why was this particularly important in Ireland? And I think it probably is something that's useful in the UK as well, is that in Ireland, patients can get treatment either side of the border. So when we look at patients that are, we'll say in Donegal, a fifth of those patients will actually get treated in Letterkenny. So when you have patients going either side of the border, they want to know what drug access is available in one area versus another. And actually, when we look at the developed nations in the UK, you also see that drug access actually varies. And you see this in Germany, you see this throughout Europe, that actually access to medicines is highly variable, dependent on where you are. So what did this require us to do? Well, we actually, a lot of the, we went through the EMA, so everything that's approved by the European Medicines Agency, these are all of the drugs that are licensed. Then we went through all of the documentation from the Irish health system. There's two different systems that we use to approve drugs. One is an older system and kind of historical approvals. And why is this important? Well, Ireland has some of the slowest HTA processes in Europe. So we're one of the slowest countries for approving drugs. Once something is licensed by the European Medicines Agency, it can take Ireland up to 2 to 3 years to approve it. And for patients, that wait is literally access to that medicine that could cure or at least prolong their survival or going into palliative care. So it really is something that's impactful. And when we looked at this, we see that basically about 65% of the medicines that are approved and licensed by Europe are actually available on the public system in Ireland. So that's a really big gap. We're now curating the NICE list, which is the UK list of approved medicines, and specifically the subset then that are available in Northern Ireland. So we can derive these All Ireland reports. And this is available publicly. This is IE Molecular Oncology Almanac, MO Molecular Oncology Almanac.org, where you can search basically the medicines that are indicated. By gene, by cancer type, by therapy. This has a full API into it, so it can actually be incorporated into, a gene ontology, gene set enrichment and thing if you want to actually, annotate maybe genes that are coming out of some spatial or some gene expression studies, or we also have pipelines for annotating VCF files and generating molecular reports. Okay, and this was basically why this is important, because we found 65% of European Medicines Agencies approved indications are actually reimbursed in Ireland. And this is causing a really big thing for oncologists, many who treat both public and private patients, and they see that they can give their private patients access to medicines and their public patients can't get this. So that's one of the things we're doing. Okay. So then the second thing that we've kind of done a lot of is trying to think, well, actually, how do we actually quantify this effect? And it's actually really, really difficult. And when you get into kind of molecular indications, you realize it's not a straightforward thing. And this is just, bevacizumab. And you see that to actually give this drug, you're giving it in combination with another drug. It's for first line treatment. And this is actually, sometimes things are not recommended for first lines. That's the first treatment a patient can get. Sometimes things are only available at second line, or some patients can have up to maybe 10, 12 lines of therapy. And this is only for patients with unresectable, advanced, metastatic, recurrent, and then non-squamous non-small cell lung cancer with EGFR activating mutations. Now, this is very, very specific. And, for bioinformatics people that are looking at, kind of treat data, we don't often go into this kind of level of detail. And if you're thinking of the annotation that's available, in a typical experiment, we don't have this. Even cancer registries don't count this. We will be able to work out who are the advanced patients, because we typically will have stage in the cancer registry. So we'll know whether the patients are stage 1 to 4. But metastatic and recurrent cancers that are not diagnosed as metastatic, that's actually a really big issue. There is a really nice project called NatCan, I think is what it is. In the UK, it's run out of London, where there's a net cancer audit, but it's like trying to identify how many patients, how many metastatic survivors there are. So patients that will have survived primary treatment and then go on to have metastatic disease, often unresectable, or the surgical status is actually not available. We don't do a really good job of classifying which are activating mutations versus just mutations. So this is actually very, very challenging. And so when we wanted to say like how many patients are not getting access to drugs, we had to make a lot of assumptions. So we took the National Cancer Registry data here to answer how many patients there are in Ireland. We looked at the ELIXIR Genie data to get an estimate of the prevalence of a somatic mutation in a cancer type. And anybody here who's working in somatic mutations in cancer will know that there's genetic variation and there's geographical variation in somatic mutations because, you know, whereas germline mutations are inherited and we can kind of predict the level of those, somatic mutations are those that you get during your lifetime. And that's dependent on the exposures that you have during your lifetime. And therefore, that's geographically determined. And there's some really nice work that like the Stratton group are doing and Paul Brennan's work have done to show kind of that. But anyhow, what we did do then is we basically did this analysis comparing the number of patients that have a disease based on the National Cancer Registry, trying to estimate how many might be metastatic, advanced, various things. And then basically from that, we could see that there was massive gaps in genetic testing being available where we don't have the information of which patients may have this mutation. And we've made recommendations to like expand the National Cancer Registry or the National Cancer Test Directory. Okay, so that's some of the stuff that we're doing on kind of national kind of infrastructure side of things. And really like a lot of the other work that we're doing is within the 1+MG, so the 1 Million Genomes program. This is a European program that is really started pre-COVID with European countries coming together to say, how do we share genomics data? And of course, in Europe, this is, you know, the GDPR legislation passed in 2018 makes this more complicated. And so sharing genomics data can no longer be in a centralized database that's EU accessible. It now has to be sovereign states and then setting up a federated infrastructure. And a lot of this grew out of then of the EGA, the European Genome Phenome Archive at EBI, and how to set that up in a federated approach. And the team in Ireland, we're an onboarding country into this, is led by John Pierre Cavalieri in the Royal College of Surgeons in Dublin. And the work that I've undertaken has been really in defining what are the variables that we need for actually doing this. And the genomicsdata.ie here is the website that we have in Ireland. And so we have 42 variables that we have defined within this 1+MG minimal data set. I'm also working on the HL7 common European common model, which is kind of the European version of MCODE, which is also defining a minimal data set. There's also CDIS, which is developing a minimal data set. When we actually look at that, there's probably about 14 different projects that are defining minimal data sets for cancer that are being adopted. So, you know, which of these will work? What way should we be thinking of all of these minimal data sets where we have, you know, we'll say, well, the Digi1 project has 37 variables. This is 42. You know, what is the minimal packet of information that we need to provide with cancer data in order that that data can then be informative. And there's a couple of different approaches to this, and I'm just going to explain those really briefly. And so this is kind of really when we begin to start thinking of federated data, sharing data across borders and real world evidence research. And this is really important when we have silos of data. And this is very much the case in Ireland, where we have data that's held locally within each site. And that means that we've fragmented access. And the UK has done a phenomenal job of developing a network of trusted research environments across the NHS, which offer phenomenal access to data locally, but doing integrated analysis across those trusted research environments, it becomes almost impossible if the format of the data in each of those trusted research environments is different, and if basically you have very different variables and very different data standards at each of those. So aligning the data standards really is critical if we want to be able to do like cross-site or federated research. And so this is a project that we're leading, and this is with Queen's University Belfast and Mark Lawler. This project started in 2022, and it's 8 clinical sites and 7 academic sites, and we've got quite a large team on this project. We have research assistants at 6 hospitals now. We're hopefully will have more hospital research assistants. And they're actually working in the clinic, because what we have in Ireland is a mixture of electronic data. We're still using paper records in some cases. And the Cancer Registry recently said that like they had 800 different sources that they pull data from. So understanding that heterogeneity of existing data sources and how we bring those together. So within this project, working to align data standards is absolutely critical. And so one of the things that we have done is we've joined a lot of European organizations and global organizations to work out, right, what are the data standards that we should be using to be able to connect this clinical data, because until we can actually define, those inclusion exclusion criteria for, access to drugs and for clinical trials, which are really, really complex and require access to the clinical data, we can't really advance a lot of the biology understanding that really has real life impact to patients. So this is just some of the I haven't actually HL7 fire here, but those are some of the organizations we're working at. I'm going to give you a whirlwind tour of those in less than 3 minutes. Okay, so GDI I mentioned already. CANDLE is EU community support action to connect cancer national data nodes, and I'm the lead of that in Ireland. JPCM is a joint action, so those are the really big EU grants, and it's got 29 countries, and it's a program that kick-started this year, and the goal is to share clinical and genomics data and imaging data, in creating kind of an ELIXIR Genie for Europe that will support ctDNA studies. It will support transnational molecular tumor boards. ELIXIR you're very familiar with. We're working very closely with an ELIXIR and the phenomenal work that ELIXIR has in compute data interoperability tools and training. And then OHDSI you may or may not familiar with, so I'm going to explain that one a little bit more. So OHDSI is the Observational Health Data Science and Informatics Initiative, and it really came out of a clinical trial that failed. So in the US, there was a medication called Vioxx that was actually a blockbuster pain drug that was widely used in the late 90s, early 2000s, around then, about 20, 30 years ago. And however, then was massive lawsuits because there was a higher rate of cardiotoxicity and heart attacks as a result of this. And when the FDA went back to look at the clinical trial data, they realized that each of the clinical partners had their data in very, very different formats. And so OMOP, the Observational Medical Outcomes Partnership, was actually an NIH grant to try to align the data across the different hospital sites. And there was more than 10 hospital sites in the clinical trial. And after that 5-year NIH grant to try to show how they could align the data across the hospitals, they realized this is really, really useful. We should continue this, and OHDSI was established to continue the OMOP common data model. And basically, what is OMOP? Well, basically, it is lots of commonly used vocabulary, such as SNOMED, the International Classification of Disease, LOINC, RX norm, ATC, a lot of these ontologies that describe clinical terms or treatments or medical procedures are then mapped onto common concepts, because you will have maybe the concept for neoplasm or advanced disease in multiple vocabularies, and what it does is it maps those onto common concepts. There's millions of concepts in OMOP, and over 140 vocabularies are mapped onto this. And this is a list of some of the vocabularies that are mapped on to OMOP. So it allows you to take data in a hospital which may be using ICD-10 or maybe using SNOMED or maybe using LOINC, and a different hospital that might be using a different version of SNOMED. There's national variations of SNOMED. And also they might be using ICD-10 rather than ICD like 11, or they might be using ICD-9, an older version of it, and allows you to map across ontologies. Within the EU, some of the largest programs in this are actually being led out of the UK. So Danny and the team in Oxford are leading this European Medicines Agency program called Darwin EU. And in that, the data is sovereign, it's in each country. And then there are 30 data partners, including the UK. And this reflects 39 sources, 16 countries, and over 180 individuals across the UK and Europe, whose data are actually mapped to this common format. And that allows you then to actually build a query in this and send that query out to each of the data partners. At the moment, this is very much done. You build an R package that allows you to do an SQL query on the OMOP common data model, because these concept terms I didn't mention are actually organized into a 39 table SQL schema. So these are actually organized that each concept is mapped to a specific domain or specific table in an SQL database. And so you can now basically, now that this is a structured SQL query or schema, you can now query that knowing where to find those terms. So you can build a query, send it out, or that could be an AI learning algorithm that you send out to each site. It's run locally and the aggregated results are returned. No identifiable information is shared. And then you perform a meta-analysis on the actual aggregated data that you get back. This is a study from HICO, which is the Irish Information and Quality Health Authority. And basically, they see that OMOP is being increasingly adopted across European countries as a framework for secondary data use under the European Health Data Standards. And it is also the major clinical standard that we've adopted within the eHealth for Cancer project that we run, where I started back, that we were trying to integrate data across 8 clinical sites. We've basically adopted OMOP as that standard. We have still to decide exactly the genomic standards and the imaging standards, and we will work all of those out. At the moment, we're doing multiple different standards, but like OMOP is pretty much the clinical standard that we're using. and we have a report on how we can actually expand that across Ireland. And as I mentioned, this is basically within the EHDS. I have one slide on the EHDS. I don't know how familiar people are, but the European Health Data Space is a European mandated legislation. So it was passed in March of 2025, and it mandates that all health data is available for both patients to see. So that's the primary access and primary care, and also for research. So that if I attend a hospital in Ireland and I'm on holiday in France, I can share my QR code. The treating physician can actually see my health records and treat me appropriately. But also my health data should be available for research in EU-wide studies. And within the EU, within cancer specifically, there's a couple of different grants that have been funded. So the European Patient Platform, aligning with the first use case there, the primary use case of data, is called EUSIP. There's the UnCan Connect platform, which is the technical one, and CANDLE is the National Cancer Data Nodes. And so there's a lot of different funding programs to be able to establish this. And then finally, where do I see this going? So if we have national cancer data nodes, such as like what we're doing in the eHealth Hub for Cancer, where we're trying to connect data across sites, have interoperable data standards such as OMOP and genomics and imaging. And then we have now national cancer data nodes with all of the data for all of the cancer patients that's available real time. We now actually have a lot of information that can underpin research. And increasingly, this data is available in the EU under programs like Darwin, but really in the US, it's really, really growing. And this is kind of what we call real world data. And this is actually a slide from Novartis. And this is the relative importance of clinical trials. So RCT here is this blue line here. I don't know if you can see my mouse. Can you? Pointer options visible. You can see the blue line. That's clinical trials. These are really, really important. This is what changes care. But clinical trials, this blue line. This will always be important. Clinical trials are absolutely essential. These are the gold standard. But increasingly, we're using these non-clinical trials or real-world data as an impetus to change care. And this is a really, really old slide from NIH. Basically, you have interventional trials, and these have always been absolutely important. But observational studies looking at the actual real world is increasingly important to changing care. And why is this? And this is because basically of the inclusion-exclusion criteria we run with clinical trials. So when we run clinical trials, we normally have, increasingly less basket, more complex umbrella studies than this, but in a typical randomized clinical trial, we have an inclusion group, and it's a small subset of people that we compare one intervention versus the other. And this could be a medicine. It could also be, a way that we actually have care. But this excludes loads and loads of people. Inclusion criteria, by default, we don't have people with complex morbidities. We don't have people that are obese or morbidly obese. We don't have people that are too old, too young, too frail. But in the real world, that's where a lot of patients are. And then medicines will be approved based on clinical trials, but we really don't know about the complex interactions between drugs or in patients with comorbidities. And so real world evidence and looking at the real world data gives us access to this patient group. So we can now look at the interactions and potentially exclusion or inclusion criteria that should be expanded or changed based on looking at the comorbidities, the frailty, obesity, children, pregnancy, all of these, because these people will have been getting medicines that are approved based on the fit people in the clinical trial. And now we can actually begin to actually really understand the real world impact of drugs. A lot of times it actually comes down to behavior as well. So in real world, in clinical trials, we're looking at that biology, we're focused on the biomarkers, but actually when it comes to practice and care and actually how those drugs are delivered in the real world, behavior is a massive thing. And this really does impact outcome. If a drug has too many side effects, people won't take it, they won't adhere to it. So my interest is very much how do we develop national and federated infrastructures so we can inform care and deliver cancer outcomes using not just bioinformatics data, but real world clinical data, and how we can develop the standards and structures for those.